Hello, everyone. I'm delighted to welcome you to what I consider the most exciting area of modern medicine, gene therapy. Today, we're not just talking about treating symptoms, we're talking about fixing the fundamental blueprint of life, the DNA itself. Think of our genome as a massive instruction manual. For people with genetic disorders, a critical page, or a single letter on a page, is missing or corrupted. Gene therapy is the technology that lets us go in and correct that error. Our lecture today will cover three core concepts. First, what exactly are we delivering? Second, and most critically, how do we deliver it? This is where the concept of vectors comes in, looking at both viral and non-viral methods. And finally, we'll dive into the serious scientific and ethical challenges, particularly the issue of off-target effects and safety. By the end of this session, you should be able to articulate how a single piece of DNA can become a therapeutic agent and why the delivery system is the key engineering challenge in this field. Let's get started. Let's start with a formal definition. Gene therapy is the therapeutic application of genetic material, DNA or RNA, to a patient's cells. But what does that really mean? It's a mechanism to address disease at its source, the underlying genetic fault. We have two primary goals here. The most common is correction. If a patient has a defective gene causing a disease like cystic fibrosis or hemophilia, we introduce a normal, functional copy of that gene. That new copy then takes over the cell's machinery to produce the missing or correct protein. The second goal is to add a new function. For instance, in cancer therapy, we might engineer T cells to express a gene that makes them incredibly efficient at recognizing and destroying tumor cells. This is the basis of sophisticated immunotherapies. The dream of gene therapy has been around for decades, but the science truly matured in the late 1990s and 2000s. Early breakthroughs in treating children with SCID, severe combined immunodeficiency, often called bubble boy disease, demonstrated that this was not science fiction. It was real, life-saving medicine. However, even those early successes came with significant risks, which we will address shortly. When we talk about the drug in gene therapy, we're talking about the nucleic acid payload. It's not a chemical molecule, it's a genetic sequence. The most straightforward payload is the corrective gene. This is simply the full-length sequence of a healthy protein-coding gene packaged into our delivery system. A more subtle approach involves gene silencing, using tools like small interfering RNA or siRNA. Instead of adding a gene, we use RNA fragments to interrupt the expression of a problematic gene. Imagine a cancer where an oncogene is overexpressed. We can silence that bad actor. Then we have specialized tools like suicide genes used in specific contexts. We engineer a patient's cells to take up a gene that, in the presence of an inactive drug, converts that drug into a potent toxin, essentially programming the cell to self-destruct. This is highly targeted cell death. Regarding how we get this payload into the patient, we have two methodologies. In vivo is direct injection. We inject the vector straight into the bloodstream or the target tissue, like the eye or muscle. Ex vivo is often preferred for immune cells. We take the patient's cells out, manipulate them in the safety of the lab, confirm the modification, expand the population, and then infuse the modified cells back in. This gives us much greater control and quality assurance. Here is the absolute central engineering problem of gene therapy, delivery. Think about it. DNA is a huge, highly charged, fragile molecule. If you inject it directly, the immune system will obliterate it instantly, or it simply won't cross the cell membrane, which is designed to keep foreign material out. We need a vehicle, a vector, that can cloak the DNA, navigate the body's defenses, find the correct target cell, and efficiently unload the therapeutic payload into the cell nucleus. The vector is arguably more important than the gene itself. We categorize these vectors into two main groups. The most effective, historically, have been viral vectors. These are essentially viruses that we've disarmed, removing their disease-causing genes but keeping their highly efficient infection machinery. They are nature's perfect delivery vehicles. The other category is non-viral vectors, which are synthetic. They often involve packaging the DNA in some kind of lipid or polymer. They are generally safer because they don't trigger a viral immune response, but they are typically much less efficient at getting into the cell compared to their viral cousins. The choice of vector is critical. It determines which cells can be targeted, how strong the immune response will be, and how long the gene will be expressed. Let's focus on the stars of the show, the viral vectors. We use them because millions of years of evolution have made them experts at one thing: getting their genetic material into a host cell. The clear frontrunner today is adeno-associated virus, or AAV. It's the favorite for a few key reasons. First, it causes very little disease in humans, meaning the immune system doesn't launch a massive attack against it. Second, it can infect both dividing and non-dividing cells, which is crucial for treating static tissues like neurons or muscle cells. This is why AAV is used in several FDA-approved treatments, notably for certain forms of inherited blindness. Its main drawback is its small carrying capacity. You can only fit relatively small genes inside. Then we have the lentiviruses, which are derived from HIV. Don't worry, they are rendered completely replication defective. The key advantage of lentivirus is its ability to integrate its DNA into the host cell's genome. This means the therapeutic gene is permanently written into the cell's operating instructions, offering long-term expression. This makes them perfect for the ex vivo cell therapies, like the widely successful CAR T-cell therapies for certain cancers. However, integration carries a potential risk of insertional mutagenesis, a point we'll address soon. Finally, adenovirus. It has a huge carrying capacity, but is highly immunogenic, meaning it triggers a massive immune response. Its use is now largely relegated to short-term, high-dose applications. Moving to the synthetic side, we find the non-viral vectors. They sacrifice some efficiency for a significant gain in safety and ease of manufacture. The huge advantage here is that they don't carry the risk of viral-related immunogenicity or insertional mutagenesis. They are just inert synthetic molecules. The most famous example, certainly in the last few years, has become the lipid nanoparticle, or LNP. This technology is behind the incredible success of the mRNA COVID-19 vaccines. These are tiny spherical liposomes, essentially fatty bubbles, that encapsulate the mRNA. They fuse with the cell membrane and dump the payload. LNPs are perfect for delivering mRNA because we often only need a transient or short-term burst of protein expression, just enough to train the immune system or, in the case of some therapies, to edit the genome before being broken down. The most basic approach is naked DNA injection, literally injecting the DNA, often into muscle tissue. This works, but its efficiency is low, sometimes requiring an electrical pulse, called electroporation, to temporarily open up the cell membranes to increase uptake. While less efficient in general, non-viral vectors are safer, cheaper to produce, and their chemical structure can be easily fine-tuned, making them an active area of research. Now we turn to the serious scientific hurdles. The first and most immediate challenge is the immune response. When we introduce a viral vector, even one that's been disarmed, the body sees the viral capsid, the protein shell, as foreign. It's a huge alarm signal. This reaction can cause two major problems. First, it can lead to systemic inflammation and toxicity in the patient. Second, and often more devastating therapeutically, the body generates antibodies against the vector. These antibodies will then neutralize the vector before it can even reach the target cells, making the therapy completely ineffective. The body destroys its own medicine. Furthermore, even if the vector gets in and the therapeutic gene starts producing the new protein, the body might recognize that new protein as non-self and launch an immune attack, destroying the very cells we just fixed. To mitigate this, patients often require immunosuppressive drugs during and after the therapy, and we must screen patients for pre-existing antibodies against common vector types, like AAV, before treatment. The second major safety challenge revolves around specificity, getting the payload only where it's needed. This is the issue of off-target effects. When we inject an AAV vector systemically, for instance, we want it to go to the liver, but a significant portion might end up in the heart, the spleen, or the brain. If the gene product is toxic in those unintended tissues, we've created a new problem. This is a vector-related risk. We are constantly engineering vectors to be more cell type specific by modifying their outer coat to only recognize certain cellular receptors. The other severe risk is specifically associated with integrating vectors like lentivirus, insertional mutagenesis. Remember, these vectors insert their DNA into the host genome to ensure long-term expression. If this insertion happens randomly and it lands right in the middle of a crucial gene, say, a tumor suppressor gene that prevents cell division, it can disrupt that gene's function and potentially trigger the development of cancer. This exact complication occurred in early SCID trials and underscored the need for better control over where the gene integrates. This risk is why we now prefer to use non-integrating vectors when long-term expression isn't absolutely necessary. Beyond the bench science, gene therapy faces significant real-world translational and ethical hurdles. First, controlling the gene expression is difficult. We need Goldilocks levels, not too low, or it's ineffective, not too high, or it could be toxic. Maintaining expression over decades is also a huge unknown. We're essentially reprogramming a cell, and we need to ensure that programming lasts for the lifetime of the cell, without the body switching it off. Second, and perhaps the most pressing societal issue, is cost and accessibility. Current approved gene therapies, particularly for rare diseases, are among the most expensive drugs ever developed, often costing millions of dollars per dose. This raises profound ethical questions. How do we fund these curative treatments? Should only wealthy countries or individuals have access? This challenge requires innovative pricing models and global policy discussions. Finally, we must distinguish between somatic and germline therapy. All current clinical trials focus on somatic therapy, where the genetic change is confined to the patient's body cells and cannot be inherited by their children. Germline therapy, which involves modifying eggs, sperm, or early embryos and would be inheritable, is ethically and legally restricted in almost every country due to the permanent, multi-generational impact on the human gene pool. We must proceed cautiously and thoughtfully. To wrap up, we've seen that gene therapy is a powerful paradigm shift, using DNA itself as medicine. The core technology lies in the vectors, which are the mail carriers that determine the therapy's safety and efficacy. We've explored the relative strengths of highly efficient viral vectors like AAV and Lentivirus and the safer, highly tunable non-viral vectors like LNPs. While we've made enormous strides, the challenges of immunogenicity, off-target effects, and high cost remain the primary obstacles to making this medicine routine. The future, however, is exceptionally bright. The field is rapidly innovating, constantly engineering smarter vectors with improved targeting specificity. Furthermore, the integration of revolutionary technologies like CRISPR-Cas9 is changing the game. CRISPR doesn't just add a gene, it allows us to edit, delete, or correct a faulty gene with unprecedented precision. Gene therapy is moving beyond rare, single-gene disorders and into complex, common diseases like Alzheimer's, Parkinson's, and heart disease. It's an exciting time to be a biologist. Thank you for your attention.